Hello, welcome once again to the new video of Premade Pro. Today we will continue with the Lac Operon and I'll try to complete the Lac Operon by this lecture. This is gonna be a bit long lecture, but try to concentrate on it because a question will come from it. 4 marks will be awarded if you get through this concept. Okay, so do not waste your time because the exam is getting nearer, and so we'll start with the Lac Operon. So before starting the Lac Operon, you must have to know that a gene must have a regulator, because to operate a gene a regulator is required. So to switch on or switch off thing, the regulator is in our hand and we operate the regulator through a operon. Okay, so what is the regulation of the gene expression? So regulation of gene expression, regulation of gene expression. In the next chapter I will start with the human nervous system. Okay, so and there is another channel coming with us. It's of a competitive examination for the government exam. I'll teach you maths. Some of you are going to appear on it because CHSL, that is the Staff Selection Commission, CHSL, the Public Service Commission of West Bengal, and different boards only requires class 12 or class 10 qualification to appear on the government job. Okay, so you can also access to that. I will give the banner and give the announcement by tomorrow. Okay, so regulation of the gene expression carries DNA changes to protein. Okay, in eukaryotes. So in eukaryote, what happens? In eukaryotes, the DNA is converted into immature RNA. Okay, DNA to RNA, RNA to protein. So DNA converts to immature RNA, that is the primary transcript. It is also called 1-degree transcript. So the immature RNA now moves to mature mRNA. Okay, so in mature mRNA, processing or splicing happens. Mature mRNA. Now the mature mRNA, then the mature mRNA is the nucleus. Mature mRNA of nucleus move to mature mRNA cytoplasm. Now the mature mRNA converts to protein. This is called the translation. So mature RNA is converted into protein and it is called a translation. So in the prokaryotes, this is going to be a part of the eukaryotes. In case of the prokaryotes, the dominant site is the initiation of transcription. So dominant site is the initiation of transcription. Now comes to the Lac Operon. The Lac Operon. Lac means lactulose. Metabolism of lactose. Metabolism of lactose. In bracket, you can say catabolism. Okay, so metabolism of lactose, that is the metabolism means breakdown. Breakdown of disaccharide. Segment of DNA which consists of gene. So gene consists of, the regulator, promoter, operator and the structure. Regulator, These four parts are the structural part of the gene: regulator, promoter, operator, and the structural. Okay, these four things generally control the total concept. Okay, these four things control the total concept. Now the lactose changes to glucose plus galactose. Glucose plus galactose. It converts to energy. Okay. So it has been named by Jacob and Monod. The Lac Operon has been named by Jacob and Monod. Jacob and Monod, the two scientists, named the Lac Operon. Okay. Now the concept. What does the concept say of the Lac Operon? The concept. So the concept of the Lac Operon. Now the Lac Operon, the concept is the absence of the lactose. Absence of lactose. Absence of lactose. Lactose needs to be absent. Okay. Now see, I'm drawing a picture. Okay, here in the start regulator gene, inhibitor, that is we can say as I gene. I gene is not an initiation gene, it is an inhibitor gene. Okay, now this is the promoting gene. Now here is the actual operon. Okay. Here it consists of Lac Z, Lac Y, Lac A. This question comes: what does the Lac Z performs, what does the Y performs, and A performs? Okay. This is the structural part. Now from the regulator we see mRNA. mRNA is converted into protein, that is active protein, and it is active protein. Okay, this protein acts as a repressor. Okay, this protein acts as a repressor. Now acts as inhibitor. Okay, now in case of Lac Y, no transcription. No transcription. Okay, no translation. If Lac Operon is off, that is if Lac Operon if Lac Operon of, that is lactose absent. Okay, completed. Now what happens? This repressor protein is completed. Now the repressor protein binds to operational group. Okay. Operator gene. The repressor protein binds to the operator gene. Now operator gene gets blocked. Operator gene is blocked. So what happens if the operator gene is blocked? RNA polymerase cannot bind to the promoting group, cannot bind with the promoting group with PG. What happens if the RNA polymerase cannot bind to the promoting group? Ultimately what will happen? RNA polymerase will not reach at the structural gene. RNA polymerase will not reach at structural gene. Okay, so the RNA polymerase will not reach to the structural gene. What will happen? 1. If the RNA polymerase cannot reach to the structural gene, what will happen? Very good question. The DNA binding site. One is the DNA binding site. And 2 is lactose binding site. Okay. Now the concept 1. Okay, fine. Now in the concept we see when the lactose, if it is lactose, it binds to lactose binding site. Lactose will bind to the lactose binding site. The lactose will bind to the lactose binding site, and the site of the repressor protein, or you can say it RP. The site of the repressor protein. Okay. Now in the second step, the structural conformational change in DNA binding site of repressor. It is the third one. So the repressor protein becomes inactive. And separated from the operated group and separate from OG. In 4, what happens? RNA poly binds to promoting group. RNA polymerase will reach at structural gene. Okay. So, did you get it? What I have said? Now, Now transcription and translation. Translation. Lac Operon on. So what will happen? Lactose inducer off to on. Lactulose is to inducer, switch off to switch on. Okay. Now happen what? Here you see let it be the beta galactose. Lac Z secrete beta galactose. Lac Y secretes permease, and Lac A secrete trans acetylase. These are asked in the Neet probable. Neet. Okay, this question can come in the exam. Okay, now so this has been completed. So write a secret point of it. What is the secret? Lactose enter Lac Operon on permease will form later. Okay. Very low level of enzyme present in bacteria. If there is a high amount of enzyme is present in the bacteria, then the lactose will not enter into the cell. So a very low level of enzyme is required. Okay. So here is one thing: a positive control of Lac Operon. Positive control of Lac Operon. Let it be the operon. It is the regulative part. Okay, this is the structural part. Now what happens? This is the cap binding site. So this red and yellow is the cap binding site. Okay. Now if glucose absent, then lactose present. Camp is formed. Cam formed. Okay, camp is formed. Now from the camp it binds to the catabolic inhibitor, the catabolic activator protein. We call it as cap. So this cap is catabolic activator protein. Okay. Now this catabolic activator protein ultimately RNA polymerase, this catabolic activator protein RNA polymerase binds to the TG, that is a transcript, it is called the transcription script. Okay, now here glucose present, lactose absent. If this is the case, no CAM, no complex formed, no complex will form, no binding to CAMP site, RNA polymerase not able to bind at promoting group, and hence no transcription. So I have completed the Lac Operon, and in the next lecture DNA fingerprinting is not required because you will read the NCERT from it, because there is nothing to understand in the DNA fingerprinting. I'll rather than provide a short video of the DNA fingerprinting, but in the next video I'll start with the human nervous system of the human physiology. Okay, so till then, good night, and please revise this long chapter, and please share and subscribe this. I am seeing that due to the long videos, some of the viewers are skipping the introduction part, the important parts, but don't skip the video. Try to watch whole video, because it is very much beneficial for your exam, because question will come from it. Okay, so bye, good night.